What if your body is breaking down a little more each day? What if every time your cells divide, DNA gets damaged, and repair can't keep up? Scientists have discovered something unsettling. Your cells have a built-in clock, and the time it keeps is limited. But here's where a crucial question emerges. Is aging an inevitable natural process, or a biological phenomenon we can intervene in? Let's explore the mechanisms of aging and the cutting edge of science attempting to slow it down. By the time you finish this video, you'll see aging from an entirely different perspective. Chapter 1. The clock written in our cells. Can your cells divide forever? Intuitively, you'd want to say yes. But the answer science gives us is no. In 1961, Leonard Hayflick discovered something remarkable. Human cells in culture divide roughly several dozen times, then stop dividing altogether. This was a startling discovery. Life had a built-in limit. Later, Elizabeth Blackburn unlocked the secret of telomeres. Telomeres are structures that protect chromosome ends, and they shorten with cell division and oxidative stress. Think of them like the plastic caps on shoelace tips. When they wear down, chromosomes become damaged, and cells age. And here's where a crucial question emerges. If telomeres didn't shorten, would cells divide forever? In fact, some cells can do exactly that. Cancer cells. While telomere-driven growth arrest suppresses tumors, telomere dysfunction can also drive cancer through chromosomal instability. Life's defense mechanism is a double-edged sword. But telomeres are only one piece of aging. At a deeper level, cells continue accumulating damage. Chapter 2. The accumulation of damage and cellular rebellion. Your DNA is under attack right now, this very moment. UV radiation, reactive oxygen species, metabolic byproducts. Every day, tens of thousands of lesions are etched into your cells. When you're young, cellular repair mechanisms fix this damage. But as you age, repair can't keep pace. And here's where something fascinating happens. Cells with accumulated damage stop dividing, but don't die, they linger. Scientists call these senescent cells. These cells are like zombies, alive, but not functioning. What's more, senescent cells secrete inflammatory factors through what's called SASP, the senescence-associated secretory phenotype. When chronic, this can harm nearby healthy cells, though in some contexts it may contribute to tissue repair. Scientists began to wonder, what if we could remove these senescent cells? In 2011, a Mayo Clinic research team succeeded in eliminating senescent cells in mice through genetic engineering. The result? Age-related pathologies were delayed. Think about that for a moment. If aging is driven by the accumulation of damaged cells, could removing them restore youth? This principle led to drugs called senolytics, some of which are now in clinical trials for specific diseases. But aging has other mechanisms at work. And one of the most promising interventions came from an unexpected place. Let's see how this discovery might reshape the human future. Chapter 3. The key to slowing aging. In the 1970s, scientists discovered a bacterium in the soil of Easter Island. A compound extracted from it, rapamycin, was initially used as an immunosuppressant. But researchers stumbled upon a remarkable effect. Multiple studies have shown rapamycin extends lifespan in mice, though the extension rate varies with dosing and sex. Still, it was a groundbreaking discovery in aging research. Rapamycin inhibits a pathway called mTOR. mTOR is a critical protein that regulates cell growth and metabolism, activated when nutrients are abundant. But when mTOR is inhibited, cells activate a process called autophagy. This is a mechanism where cells clean themselves, breaking down and recycling damaged components. In other words, rapamycin mimics a, quote, starvation state in cells, promoting repair and regeneration. And there's another promising molecule, NAD+. NAD+ is a coenzyme essential for cellular energy metabolism, and changes in NAD metabolism with aging have been reported in both animals and humans. In 2016, a study reported that long-term administration of NMN, a precursor to NAD+, alleviated age-related physiological decline in mice. However, while human studies show NMN and NR can increase NAD levels, established clinical effects on health span are still being verified, with challenges in standardizing study design and evaluation metrics. Let's think about what this means. If we can optimize cellular metabolism, repair damage, and remove senescent cells, aging might not be an inevitable fate, but an intervenable process. But here's where an ethical question emerges. If humans could live 150, 200 years, how would society change? Would resources suffice? Would intergenerational inequality expand? Intervening in aging isn't just medical progress. It forces us to reconsider the very meaning of human existence. Science has opened a door of possibility. But what lies beyond that door, we still don't know. Chapter 4. The World Revealed. Today, we've deeply explored the mechanisms of aging and the frontiers of science attempting to slow it. We've seen that aging isn't an inevitable fate, but a complex process occurring at the cellular level. Let me summarize the world that's been revealed through today's exploration. First, aging progresses through telomere shortening, DNA damage accumulation, and increasing senescent cells. Our bodies sustain tens of thousands of lesions daily, and repair mechanisms try to fix them, but fall behind as we age. Telomeres are clocks counting cell divisions, and when they shorten, cells senesce. Second, senescent cell removal through senolytics, autophagy activation via rapamycin, metabolic optimization through NAD plus supplementation. These interventions have shown lifespan extension in animal studies, but clinical effects in humans are still being verified for specific diseases and functional metrics. Aging isn't a single mechanism, but an intertwined phenomenon of multiple processes, each with potential for intervention. Third, if humans could live 150, 200 years, it would be both medical success and a source of social and ethical challenges. Resource allocation, intergenerational equity, and the very meaning of a good life would be reconsidered. Current medicine and regulation don't define aging itself as a treatment indication. Interventions are verified as improvements in specific diseases or functional metrics. What these insights reveal is that aging isn't an inevitable fate, but an understandable, intervenable process. Through today's exploration, I've felt anew. Aging is a journey life takes through time, and that journey holds dignity and meaning. But if we could extend that journey, how should we live? Science shows possibility, but the choice remains ours. This is a question that inspires awe. So I ask you, if you could slow aging, what would you do? And if life were 200 years long, would it hold the same meaning as your life now? These questions have no easy answers, but continuing to ask them moves us forward. After finishing this video, when you return to daily life, I want you to be conscious for just a moment. When you see wrinkles or gray hair in the mirror, they're not just traces of aging, but results of complex processes happening at the cellular level. And science is unraveling those mysteries, exploring possibilities for intervention. Aging isn't an unavoidable process, but a phenomenon to understand and confront. Share your insights in the comments. Your questions are the force that drives this channel forward. Thank you for joining me on this intellectual journey to the very end. Today we explored aging, a universal process we all face. This channel continues exploring every question, from the frontiers of science to the essence of human existence. If you found value in this exploration, subscribe and join the next intellectual adventure. Your curiosity is the force that propels this community forward. Until next time.